our last installment on carbohydrates, going to cover the functions of glycoconjugates. So let's talk about what a glycoconjugate is. It's when you conjugate, okay, or you're going to add or covalently link, okay, something to a glycan, right? So we've got a carbohydrate portion, and it's going to be linked to another type of molecule, okay? So we got a carbohydrate linked to another molecule, and it can be a protein, it can be a lipid, and we'll talk about a few examples of that. Okay. All right. So the difference between when we call it a glycoprotein or a proteoglycan is which part of the molecule is there more of. So a glycoprotein has more protein, and a proteoglycan has more glycan. Okay, so it's got more sugar versus more of some other molecule, protein, lipid, whatever. All right, so in this case, the bulk of our molecule is going to be protein for glycoprotein. And these wind up being transported by like membrane vesicles or targeted to cellular organs. Whereas proteoglycans, okay, the bulk of the molecule is the carbohydrate, okay? And these are going to be ones that are located in what we call the extracellular matrix. And they're going to be covalently linked to aminoglycans. All right, so let's look at some of the monomers that are found in glycoconjugates. And I'm going to remind you that all of our monomers can be represented by one of these symbols here that are shapes. Okay, so the glycoconjugates are going to combine both modified monosaccharides and unmodified ones. So unmodified would be, glucose would be an example where it's just our CH2ON carbohydrate structure. And something like N-acetylmuramic acid, where we've got our N-acetyl group, but we also have a carboxyl group added on. So we've got functional groups added on. Those are our modified ones, okay? And again, there is an organization that it's IUPAC-like. It's called the Consortium for Functional Glycomics, CFG, kind of sounds like a spy organization, that determines who gets to be the blue circle and who gets to be the orange star. All right, so how these symbols get used are shown in this figure. And so this shows you a protein, which the protein is kind of off the screen, and it's linked to this glycan group. So we could show it, in this structural form right here, where, the only kind of shorthand that we're using is we were not showing the hydrogens and the carbon or the symbols for the carbons. We could show it in terms of the name of the monomer, okay, and you'll notice that in these names, and I'm going to zoom in a little so it's easier to see, the names are separated by the type of linkage that there is. Now you'll notice one thing about the linkages, and that is that you only see one number there, okay? And the reason that you only see one number is because this is assuming that you know something about carbohydrate chemistry. So the number, okay, is that is shown here, okay, is only the number of the non-anomeric carbon that's attached, okay, in the bond. Okay, so only the number of the non-anomeric carbon is given. And why is that? That's because when you're communicating your structural information this way, you're speaking to other biochemists who know that the other partner in the glycosidic bond has to be the anomeric carbon, and who can look at, let's say, a galactose unit, and will know how to find the, so if we look over here, here's our galactose, right? And we know that in galactose, the anomeric carbon's number 1, and that has to be the one that's involved in the glycosidic bond. So if we're taking this unit here, galactose B4 glucnac, okay? So here's our galactose unit, okay? Here's the anomeric carbon, so this is our gal. And then our glycnac, this is an acetylglucosamine, so let's make that in green. Okay, so here's our N-acetylglucosamine. The anomeric carbon for that is over here, so this means this is 2, 3, and here's carbon 4. Okay, so we know that this is a 1 to 4 bond, right? And again, when we look at this, okay, this is it's given as beta. Here is, whoops, this is the CH2OH. And let me just do that in pink so that we can, here's the CH2OH. So here's my C6, right? So that's the C6. C5 is the one that's in the ring. Okay, that is above the ring, even though it's involved in the glycosidic bond to that end unit there. So that means if this is up and that's up, they're on the same side of the ring, so that's a beta linkage, okay? So this linkage is our beta 1 to 4 linkage, okay? So again, we're limiting the information we give to tell you only what you need to know to figure out the structure if you know something about the biochemistry of carbohydrates. All right, so now we'll look at how this is shown with the symbols. So again, this even is a little bit too much, and so we use the symbol nomenclature here. So one of the things that is sometimes included in the symbol nomenclature is the bonding information. So, you know, that would be shown over the bond. So we'd have beta 4, alpha 6, this one's beta 2, and then alpha 6. Okay, so sometimes that information, the linkage information is provided because that is important. That you can't figure out from the symbols, okay? All right. So that's using the CFG symbols to represent, you know, this complex thing. And it's particularly helpful, again, when we're communicating and we're trying to figure out sort of differences of things and patterns. It's easy to, you know, it's easy for a human to look at something with colored shapes and deduce a pattern that might be harder when you have sort of more details in your image. Okay, so you know these things don't just happen. These linkages, that we have enzymes that are used to produce these structures that are linked to glycoproteins. So again, in these, you know, these reactions, it's glycosyl transferases that are the enzymes that add the units on, right? So we've got here, they're specific for like the monomer that's going to be added on. And this is how we form both the initial link to the protein, which is shown in this first step, and then how we add monomers on as we're building the thing. But you'll notice, okay, that the sugar unit that's being added on is carried there, okay, by a nucleotide that's been conjugated to the monosaccharide. So this is uridine. So this is uridine diphosphate galactose. So that's how it enters. Okay, so, and then that's how you get the reaction being added. Okay, so one of the kind of important things about these enzymes, okay, is that there are genetic differences, okay, in expression between individuals of these enzymes. Okay, these enzymes. And also the activity, okay? So you can have a mutation that affects the activity of the enzyme, how good it is at catalysis, right? And these genetic differences are the sources of immunological incompatibility, okay? So this is the source of immunological either compatibility or incompatibility. Oops. I spelled it two different ways, didn't I? That's all right. Between individuals. So what do I mean by that? Whether you can be a blood donor to someone, whether you can be an organ donor to someone, okay? And the reason that these enzymes are important in that, okay, is because they're going to control the oligosaccharides that get displayed on the molecules that are, on the surfaces of your cells. For example, we'll talk about red blood cell compatibility, okay? And because we have such a great diversity in our monomers, okay, this is why what our theme is with carbohydrates. They can be used for very specific interactions. So they can be used to tell self from non-self. And the way that you get differences in what carbohydrates get on to your surface that you're using to identify self from non-self is all in the interaction and the activity of those enzymes. So those enzymes are really important in like a medical context. Okay. All right. So that's how proteoglycans get made. What do they do? How do they function? Okay, so proteoglycans are involved in protein binding activity. So protein recognition. They also regulate signal transduction and the movement of cells from one place to another. So I think a key point here is that they're found, on the cell surface, and what am I doing here? They're found on the cell surface and outside of cells, and again, they're really important in recognition. Okay, and again, diversity of monomer leads to specificity of interactions. Specificity of interactions is important for recognizing self versus non-self, recognizing a particular function of a cell. And so we've got, all sorts of things involved in the extracellular matrix and also on the cell surface. Okay, so you'll see we've got core proteins. Sometimes they'll have our lubricating molecules that are attached to them. Sometimes they will have they'll have specific interaction glycoconjugates connected to them, and it all plays an important role. Okay, so one type of one type of proteoglycan that we haven't talked about are the peptidoglycans that are found in bacterial cell walls. Okay, so if you recall way back when we were starting to study proteins, we talked about proteins using mostly L amino acids, except in bacterial cell walls where they use D amino acids. So this is the case where that comes into play. Okay, so peptidoglycans, okay, are found in bacterial cell walls, okay, and they contain a peptide part and a glycan part. Okay, so this is the thing that's going to give bacteria their shape, and they're going to be the physical boundary between the outside and the bacterial membrane. Okay, and so what are they made up of? They've got, this is the glycan portion. Okay, so this is the sugar portion. And that's a sort of a repetitive disaccharide, usually, NAM and NAG. And then there is the peptide portion. So this is a peptide cross-linker. And it's made of D amino acids. So this is, again, that exception where we use D amino acids. Okay. All right. And we have this kind of repeated structure, okay, and a key thing from a bacterial perspective, okay, is that is forming that linkage, okay. So that linkage, okay, is made by this transpeptidase linkage enzyme. Okay, so that's going to make those peptide linkages, okay, between your chains here, right? And so this is useful from a human perspective because we have antibiotics, okay, that block the synthesis of this cross-linker. Okay, so this cross-linker right here, okay, penicillin prevents that from being made by inhibiting that enzyme. Okay. All right. So now we have these two things called gram-positive and gram-negative bacteria, and their cell wall is a really important difference between the two. So that's why we're talking about it here. Also has some interesting sort of medical connotations for us. So the cell wall in gram-positive has a really thick peptidoglycan layer. So if we take this as our zoomed in version, okay, as our peptidoglycan, and if you have many layers of that, so that's what this figure down here is. It's so each one of these is a single peptidoglycan layer. And so we've got multiple layers, layer upon layer of peptidoglycan here. Oops. Just make it. There we go. So there's our layers. Okay, so that's what we would call a gram-positive cell. Okay, that their cell wall has that type of structure, whereas a gram-negative cell only has one thin peptidoglycan layer. And it also has an outer membrane. Okay, so here is the cell membrane in a gram-positive, and here is the outer membrane, and here is the cell membrane in a gram-negative. So these two different classes of bacteria have different profiles in terms of pathogenesis. So the gram-positive ones tend to have toxins that will act on the GI tract that will give you things like let's see, so gram-positive are going to be things like staph infections. So MRSA are drug resistant. So because they have a lot of layers of peptidoglycan, these are really the targets of penicillin class drugs, right? Because they have a lot of peptidoglycan layers. So you'll notice things like E. coli and salmonella, the things that are associated with food poisoning tend to be gram-negative bacteria. These tend to be more drug resistant, even though, MRSA is the thing that we mostly think of as our classic antibiotic resistant bacterial strain. This outer membrane is what protects gram-negative bacteria from the current antibiotics that we have. So that's, again, how cell walls, are found in bacteria that have that peptidoglycan layer. And just a very quick look at that transpeptidase enzyme that I said that penicillin inhibits. Again, it's going to be the thing that creates the link between the two tetrapeptides, if you will, in the different layers of the cell, a different layer in the cell wall. All right. So, and the way that drug resistant bacteria attack this, okay, is so penicillin, okay, is a structural analog. Penicillin it looks like the natural substrate, right? It's got a beta-lactam ring that looks very similar to the natural substrate, okay? And so it can be an inhibitor of this enzyme because it can bind in the active site, okay? Now, what the antibiotic resistant organisms do is they produce an enzyme that's going to break down that ring and render this an ineffective inhibitor. Okay, so resistant bacteria make an enzyme that breaks that ring. Okay. Oops. Okay, so this is just a little bit of more detail about that. Okay. So we've already looked again at proteoglycan structures. That's a repeat slide. So I'm going to actually, while I'm here, I'm going to delete the repeated slide because we don't need it. There we go. Okay, so now let's finish up our discussion by talking about glycoconjugate function in specific recognition processes. Okay, so those are going to be cell signaling and immunity, but again, both of these fall into specific recognition function category. Okay, so we can have two types of binding that involve these glycoconjugates. Okay, we can have intrinsic, okay, or extrinsic. Okay, so intrinsic binding is going to mediate cell recognition, okay, on immune cells, and it's going to do things like helping with cell attachment, cell migration. So we're not going to talk in detail about the immune system, but one of the things that happens is the immune system has both humoral immunity, which is goes through a liquid medium, and cell-based. So in cell-based immunity, like cells of the immune system have to go from here to there to the site where they have to do their job. And so intrinsic binding is going to be involved in those processes. And then extrinsic binding is going to occur between immune cells. It's going to be involved in invading pathogens, and it's going to protect host cells from infection. Okay, so this gives you a better idea because it gives you a sort of visual examples of these types of interactions. Okay, so intrinsic recognition, okay, is going to be things like we've got a host cell that two, let's say maybe receptors on the host cell are going to interact with one another, and they're going to do that via the glycan part of your glycoconjugate, okay? Or, okay, it's going to help cells stick together, okay? So that would be intercellular, and it's going to use, okay, the same thing. It's going to use a glycoconjugate as the recognition thing, okay? All right, so this is all, recognition is all within the organism, okay? And extrinsic recognition, okay, is going to be between, okay, a pathogen, okay, and either a host cell. So this is going to be binding to an external invader, let's say. Okay, so in this case it's shown it's a little bacteria here. Okay, so here's our pathogen cell, and here is the cell of our organism. Okay, now you'll notice the word lectin is being used here. So a lectin, okay, is not a glycoprotein. Okay, that's first of all what it is not. It is not a glycoprotein. It is a protein. It is a protein that binds oligosaccharides. So lectins recognize glycoconjugates, and that's important vocabulary to know. Okay. All right. So again, they're going to play important roles in recognition. So intrinsic recognition, again, is going to be within a cell or within let's say this is a human cell over here, okay, and another human cell. So let's say this is, I don't know, a liver cell, and this is a cell of the immune system, like a T cell, okay? So there are receptors, there are lectins on the surface of our immune cells that can tell our liver cells apart from things that are not our liver cells. And this is where, again, if you're transplanting an organ, you have to make sure, okay, that the glycoproteins on the surface of the donor organ are compatible with the immune system of the person who's going to be the recipient of the organ. So this is really like glycans and glycoconjugates play an important role in immune matching for things like organ transplantation or blood transfusion. And then our extrinsic, again, bacterias, viruses, if there are incompatible proteins. All right, they're gonna bind there and recognize. Okay. N and O link like oligosaccharides. We're just going to talk about this for a second before we talk about blood grouping, and that's our last topic. Okay, so the N and O linked oligosaccharides are just whether you're linking to a hydroxyl group like you would on a serine, or you're making an amide bond like you would to an asparagine. Okay, so that's the only difference. So N-linked and O-linked, it just means what's the partner in your glycosidic bond when you're attaching a sugar to a non-sugar. Are you attaching to an amine? Are you attaching to a hydroxyl? Okay, that's all that is. And what you'll notice is that there's a specific group, this glycnac with 3 mannoses, so 2 N-acetylglucosamines and 3 mannoses. Okay, so that's all N-linked glycoproteins are going to contain this group that's shown here. Okay, so the blue squares are our 2 glycnacs, and the mannoses are our 3 green spheres. Now you'll notice that as long as you have that central unit there, okay, you can add on other things. You can add things to your mannose tail, or you can add things on to that basic core. But you have to have that basic core for all N-linked glycoproteins. And for your O-linked glycoproteins, you just need 1 N-acetylglucosamine group. So it's just that one, and you can attach to a serine or a threonine. Okay, so N-linked are going to go to an asparagine. So these are our N-linked, and those go to asparagines. And then O-linked can either be a serine or a threonine. Okay, and those are our hydroxyl-containing amino acids. We don't really use tyrosine for linking up, even though it has a hydroxyl group. We don't link up to glycans that way. Okay, so our final example that we're going to use, okay, of specific recognition is the ABO blood groups. Okay. All right. So human ABO blood groups are determined by the expression of one, both, or neither of 2 specific enzymes. Okay. So the reason you have different blood groups, okay, is because of the enzymes that make the glycoconjugates. So we're going to say linked to the expression of 2 enzymes. Okay, you don't need to know their names, but those are the ones that are responsible for attaching a sugar to what we call the O antigen. So that's the important thing, okay? It's that the difference is the expression in the enzymes that link the glycan to the O antigen. And the O antigen, okay, is present on the surface of red blood cells. And I'm going to use RBCs for red blood cells there. Okay, and notice that we're using the symbol nomenclature for our different type of our blood types. Okay, so here's the enzymes: the GTA enzyme, the GTB enzyme. Okay, and so you can either have none of the enzymes are present. Okay, that's the case when you have O blood, right? So this is the basic core that everyone's blood cells have on it, on their surface. Okay, so nobody is going to have an immune system that reacts to that. Okay. Now, if we have this GTA enzyme, we will get a modification that adds, okay, an acetylglucosamine to that O antigen. Okay. If we have the GTB enzyme, okay, instead of an acetylglucosamine, okay, what we're going to have is we're going to have a galactose there. Okay. And if you possess both enzymes, some of your molecules will have galactose and some will have an acetylglucosamine. So you will have both of those. Okay. So what this means is you will react to anything that doesn't look like something you have in your blood. So if you have type A blood, I have type A blood, okay? And so I can get blood from an O donor because the O donor has the same core as I do, okay? I cannot give blood to somebody with type O blood because I have this N-acetylglucosamine up here that they don't have, and their immune system's going to react to that. Okay, I can give blood to an AB type person because they've got stuff that looks like what I have. They cannot give blood to me because they have the B antigen. Okay, so it's all based off of, you know, whether your immune system is seeing something different than it knows itself to be. Okay. So this is just a table that summarizes the different blood groups, okay, and the compatibility. So we went over, you know, I'm type A, and I can get blood from another A, or I can get it from an O, and now the difference here is there's a difference between plasma and red blood cells. So you can get plasma from somebody who has a type AB blood. That's all related to what antibodies would be found in your plasma. Okay, so the antigens on your red blood cells are determined by, again, what one of those enzymes that you express. Okay, the antibodies that you produce, okay, are going to be in your plasma. So if you don't have, an AB donor, so here's my type AB, they don't have any antibodies in their plasma. And plasma is like the cell-free portion of blood, so they don't have any antibodies to my A, so I can do that. And the reason they don't have any antibodies to A is because they've got A, right? So that's how blood typing works, and that's where we're going to stop for our coverage of glycoconjugates. So that's it.